All right, our final speaker for the evening is Hersh Bhargava. Hersh is in the biophysics program. His advisors are Wendell Lin and Hannah El Samad. His title is entitled Reprogramming T Cell Communication to Enhance Cell Therapies for Cancer and Beyond.
All right, thank you very much. Good evening, everyone. It's a pleasure to be here. I'm excited to share new insights into how immune cells communicate with one another in the body and how we can re-engineer immune cell communication to create better therapies for diseases like cancer. The human immune system is a phenomenal piece of technology. It's been refined and developed for literally hundreds of millions of years with the sole goal of keeping us safe from disease. These diseases include not just diseases that come from exogenous pathogens invading our bodies like viruses and bacteria, but it's also the job of the immune system to keep us protected from diseases that arise from dysregulation of our own tissues, like in cancer, autoimmunity, or even degenerative disease. These cells that make up the immune system, like the T cell I'm showing here, are endowed with capabilities that are not seen in any other cell types in the human body. They have the capability to patrol throughout the body, literally from head to toe, searching for signs of disease. And in addition, they're quite literally licensed to kill. Cells like T cells, upon recognition of target cells that they deem to be potentially dangerous to the greater organism, are able to kill those cells with single-cell precision, something which can turn incredibly dangerous in the context of autoimmune diseases where that killing functionality gets mistargeted against our own healthy cells. But by the same token, this killing activity of immune cells is what keeps us safe from diseases like cancer and infectious disease. We now know that even in people who don't manifest cancer in a clinically significant way at any point in their life, they develop cancerous cells many times over the course of their life, but those cells are destroyed by the activity of cells in the immune system like T cells. So this is really important. The immune cell, interestingly, operates through the collective orchestration of literally billions of cells that exist in the body. What's really interesting about this is that it resembles something like an army or almost organization of cells. But unlike any human army or human organization, there's no hierarchy or centralized command structure. There's no generals in this army that can direct the motions of hundreds of thousands of immune cells or tell them what the bad guys look like. This is an army that's composed entirely of foot soldiers that operate in a completely decentralized manner, all operating by the same rules, each operating based on only what that individual immune cell can see in its immediate vicinity. This creates something of a paradox which I was really interested to investigate in my PhD, which is how can immune cells operate in a way that gives rise to this incredibly balanced response and incredibly sophisticated capabilities despite not having centralized architecture. And much of this comes down to how immune cells communicate with one another. And it turns out that immune cells communicate with each other by releasing molecules called cytokines into their environment, which they can also sense. And this enables a lot of different behaviors, some of which we've heard about even today. But I'm going to zoom in on T cell communication and how that gets hijacked in cancer. Interestingly, T cells, in response to the detection of target cells that they perceive as foreign, begin to release cytokine molecules, in this case, a molecule called IL-2. And as more T cells detect more foreign matter or dangerous cells in the body, the concentration of this molecule increases in the local vicinity. And when it breaches a certain threshold, that triggers a positive feedback loop where the cells collectively scale up their aggression against the threat. This enables balance such that no one T cell can go rogue and call in a dramatic immune response on its own, but literally constitutes a sort of voting mechanism by the immune system. Interestingly, in this election, if as it were, there are also pacifist cells. There are regulatory cells whose job it is to prevent off-target immune responses in autoimmune disease. These cells also vote in this election by sucking up the molecules that T cells are releasing and reducing the amount that accumulates in a local vicinity. This contributes to the balance of immune responses in healthy people, but this is something that becomes hijacked by tumors and results in tumors shutting down immune responses that would otherwise destroy them. So in a tumor where T cells might begin to recognize the tumor as foreign, some tumors will recruit those suppressive pacifistic immune cells, convert them to protect the tumor itself, and suppress T cell responses. This is something that we see as a hallmark of some of the cancers that remain the most difficult to treat and the least responsive to even the most cutting-edge cancer therapeutics. Fortunately, however, we live in the era of cell engineering, where we're no longer limited by the factory hardware and software that we're born with. We have the ability now, this is a clinical reality at hospitals like UCSF, to isolate immune cells from patients, genetically engineer them with new programming that improves their specificity for diseases like cancer, and then reinfuse those immune cells into patients' blood, where this strategy called CAR T cell therapy in its earliest form has revolutionized the way we treat certain cancers, mainly blood cancers. So we asked whether there's something we can do to re-engineer T cells in a way that would enable them to circumvent these tumors that hijack immune communication, perhaps engineer a new and more powerful communication channel into these immune cells. We looked at a preclinical mouse model of pancreatic cancer, one of the most insidious cancers in existence and one of the most difficult to treat. In this disease model, even the CAR T cells I just mentioned are ineffective in clearing these tumors, whether in mice or in humans where it's been tested. What we see is even when we inject patients with hundreds of millions of these engineered T cells, very few accumulate in the tumor microenvironment. The tumor has developed the ability to keep those immune cells out and shut them down if they do arrive in the tumor. So what we developed was what we call a synthetic cytokine circuit that supercharges the ability of our engineered T cells to vote in favor of scaling up the immune response in the tumor. This circuit enables T cells to sense that they are in the vicinity of the tumor and in response induce a very, very high level of cytokine production, these voting molecules that immune cells produce, about 5-fold more than any T cell can naturally produce, and also for substantially longer time period. This not only tricks the T cell doing the voting into thinking that there's a huge number of T cells that are activating, but it can also act on other T cells that are not engineered but are nevertheless in the vicinity and enable them to scale up their responses as well. This strategy was effective in the preclinical model of pancreatic cancer I mentioned in completely changing and remodeling the microenvironment inside these tumors. Here you can see in the brown stain are the T cells which are now infiltrating and proliferating within the tumor microenvironment. The survival data from these mice indicates that we basically saw that the circuit, the synthetic cytokine circuit, enabled the complete clearance of tumor from all the mice in the test group versus all the mice in the that were treated with conventional CAR T cells succumb to the disease. So in my graduate work, I'm continuing to explore what is a very complex and sort of sophisticated natural regulatory network of how immune cells talk to one another, which I've spared you the details of. But it's really the fundamental idea I want to leave you with, that by making basic science discoveries about how different systems are implemented by evolution and how immune cells behave naturally, now in this era of cell engineering, we can take those basic science insights and then convert them into new strategies for treating diseases. And that's exactly the kind of work that the Discoveries Fellows Program supports, and I'm delighted to be a part of. So with that, I will thank my advisors, colleagues, collaborators, and the Discovery Fellows Program for your support, and all of you for listening. Thank you very much. And I'll take a question. That's a great question. So it has actually a very old idea at this point that could we administer this cytokine IL-2, the question was how effective is exogenous IL-2? So could we just inject this cytokine into the bloodstream? And this was actually tried in the 80s by scientists at the National Cancer Institute. And it actually did show some efficacy in metastatic melanoma and renal cell carcinoma. But the real issue is that it's incredibly toxic. IL-2 is cleared very, very quickly from circulation. So the doses required to achieve a sufficient amount of IL-2 delivered to the tumor are just incredibly high. It's delivered intravenously. Basically, a huge amount of IL-2 is delivered, and that leads to significant toxicities and other issues, systemic inflammation, vascular leak, and so very few patients tolerate it, and it's almost never used today. So that's one of the other reasons that when we deliver it using a cell, we can get it to exactly where it's needed, hopefully, and at exactly the concentration it's needed. All right, thank you very much.
Well, there you have it.